Hello, everyone, and welcome to today's broadcast, How to Utilize the Power of CRISPR in Your Research. It is presented by Ania Wronski, Ph.D., the Product Manager for Engineered Cells at Synthego. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Synthego. For more information about our sponsor, please visit www.synthego.com. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located in the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Wronski. I will now turn the presentation over to her.
Thank you, and it's my pleasure to be speaking to the LabRoots audience to really help people understand how to utilize the power of CRISPR in their research. So what I would like to go through today is really go through what CRISPR is and what it can do for researchers today. It's an incredibly powerful tool. And then also go through what is required to have a successful CRISPR experiment, which is obviously integral to using this tool in your research. So I'll go through how to design an experiment, how to edit, actually conduct the editing, and then how to analyze the results at the end of that experiment, which is obviously pivotal to knowing whether it worked properly or not. So what is CRISPR? CRISPR actually stands for Clustered Regular Interspersed Short Palindromic Regions. Try not to say that too fast. And so this is a really powerful technique that, as of a couple of years ago, has become pivotal to a lot of how we're doing research today, and is really helping to advance the way that we can conduct research and the kind of insights we're able to get. And so what does CRISPR actually do? CRISPR can precisely change DNA sequences, and this is very different to how we've been able to modulate gene expression using other tools such as RNAi. So CRISPR uses two different complexes, what we call the guide RNA and the Cas9 protein itself. And these two complexes come together to form a ribonuclear protein, which when it goes into the cell, finds homology to that guide RNA sequence, which we designed to target part of the genome. So once that complex has found that DNA, the whole RNP complex, it will make a double-stranded break. And that is all the CRISPR-Cas9 system actually does. Once it makes that double-stranded break, obviously that's in the genomic DNA. That's not something the cell wants to put up with. And so the cell will try to repair that using a process called non-homologous end joining. And this is whereby the cell will try to put insertions or deletions within that cut site to try to repair it so that the cell can go on and still be living and stable. And so using this type of that double-stranded break, we can cause a protein knockout. So how do we actually do this using CRISPR? So if we remember back to our central dogma of genetics, our DNA, the genomic DNA gets transcribed into mRNA, which then gets translated into protein. And so if we make an insertion or deletion at the genomic DNA level, that's then going to affect the open reading frame that the mRNA makes, and then thereby the protein sequence that is utilized. And what typically happens with CRISPR-Cas9 when we get those insertions or deletions created is that we change the open reading frame that the mRNA utilizes, and then that means that we get a premature stop codon from the protein level, which causes a truncation of protein and usually nonsense-mediated decay to then have a knockout of protein function. So using non-homologous end joining, we can get that complete protein knockout. But then there's also another side of CRISPR that we can utilize. And so this is where we can use a donor template in addition to the Cas9 and the guide RNA to do more precise genome engineering. And so this is where we can use that donor template for when that double-stranded cut is made. It can then more precisely fix that double-stranded break by inserting or deleting precise pieces of DNA, which we stipulate on that donor template. Now, one thing to note is that for homologous recombination, or that process of knock-in and inserting or deleting precisely pieces of DNA, that's a very inefficient process for the cell to take. So in terms of the kinds of editing efficiency that we're able to get for knock-ins using homologous recombination, we often get very low editing efficiency. So double digits is usually a very favorable outcome. Whereas for knockouts using non-homologous end joining, we're able to get much higher editing efficiencies, and we're able to achieve upwards of 90% depending on the guide and the cell line. And so it's really important that you're aware of that, because it still is a challenging technique to do a knock-in. So how can we use this entire system for use in your research? So CRISPR is obviously a very powerful technique. Using non-homologous end joining or just making knockouts, we're able to do a lot of different things, such as study the protein function using a knockout to do a loss of function experiment. We can use knockout cell lines to validate your antibody or your assay to ensure that they're binding or functioning correctly. And then we can also use it for pathway analysis to identify which parts of a pathway is integral to the function that you're looking at. And then using knock-ins or homology-directed repair, we're able to do things like disease modeling, where we can insert SNP or codon changes to reflect disease states. We can endogenously tag proteins, which was previously very difficult to do without CRISPR. And then for other things, we can more precisely insert or delete pieces of DNA, which obviously has a wide range of applications. So how does Synthego as a company approach the way we do CRISPR? And we do things a little bit differently to the vast majority of life science companies that are out there. And we really apply the concepts of engineering onto how we do biology. So what does that actually mean? It means that we use the facets of design, build, test, and learn to really iterate on how we're doing biology. And we're placing this on top of genome engineering. So this means that we do things in parallel. We try to rapidly iterate on how we do things. We try to do things at scale, as well as utilize automation to help us be able to do all of these different things. And we apply this on things like how we build things, so the hardware that we utilize, the bioinformatics tools that we're using for CRISPR-Cas9, as well as the chemistry that we're building to be able to make better reagents, for example. And then we utilize all of this to be able to improve how we're analyzing biology and, for example, how we're making engineered cells. And so we utilize all of these different things to be able to combine them together into what we call the full stack of genome engineering. So this is really, in sort of engineering terms, the full workflow of how you can do a CRISPR-Cas9 experiment. So this includes how you design the experiment, how you actually do the editing, and then finally how you do the analysis. And so we combine all of these into what we call the full stack solutions. And so we have CRISPR Revolution kits, where we can make the synthetic chemically modified guide RNAs for use in your own cells. We provide already edited cells within our engineered cells portfolio. And then we underpin both of these different products with our bioinformatics tools, which I'll talk about in a little bit, just to really reiterate how we can provide that full stack of genome engineering solutions. And we really integrate all of these into that one product line of engineered cells to be able to get the very best editing outcomes. And I'll show you some of that as I go through this presentation. And so from that design, edit, and analyze, what we really tried to do as a company is identify where people were getting stuck. Where are the difficulties that people find in doing a CRISPR experiment, and how can we improve them for the entire scientific community so they can really utilize this fantastic tool? And so we've identified multiple different points, and I'll just touch on a few of them. So this includes, how do we build more effective reagents to do CRISPR experiments? How can we optimize to a greater degree so we can get better success rates? How do we analyze these experiments in a more high throughput and informative fashion? And then how can we as a company be able to provide solutions that are flexible and affordable to the scientific community? So starting off with effective reagents. So we designed our own design tool so that we can better design guide RNAs, which are obviously integral to how we do a CRISPR-Cas9 experiment. And so our design tool takes into multiple different facets. So we look at the early coding region of any gene when we design our guides, because as I showed you previously, we want to ensure that when we're designing a guide, we're hitting an early exon so that we can knock out that protein function as early on as we can so we don't get a protein transcripts created. In addition, we try to find a common exon. For many genes, there's multiple transcripts that are coming off that particular piece of DNA. So we want to ensure that when we design a guide, we're able to target every transcript. In addition, something that's very important for CRISPR editing is being able to control the off-target effects. So one of the ways we can do this is through the design of the guides. And for any engineered cells product or project, what we try to do is include as many mismatches as we can so that that piece of guide RNA that we're using doesn't have very high affinity elsewhere in the genome. And so we often try to do at least 3 mismatches to elsewhere in the genome to ensure that we're minimizing the off-target effects as much as possible. We also do this with our reagent design, which I'll touch on in a little bit. And then finally, we try to find guides that have a high activity. And so there's multiple different scores that people use to really try to rank guides and identify, based on sequence, what kind of guide will be predicted to work better. And from our computing power and from the ability to automate a lot of the processes that we have in-house, we're actually able to test some of these assumptions. Does an on-target score actually equate to better editing efficiency? So we actually did this experiment in-house, and using thousands of different guide RNAs and looking at the 5 different algorithms that are usually used for on-target scores, as you can see from this data, where we have the predicted editing efficiency on the x-axis and then the observed on the y-axis, you can see that there's actually no correlation for any of these algorithms between the predicted score and then the actual score. And so what this means is that currently as a field, we don't have a great way of predicting editing efficiency just from the guide sequence alone. There's obviously a lot of other complexity that is going into this that is affecting our ability to edit at any specific location. The other side of that is that we need to try multiple guides for any one gene or location, because we don't have good ways of predicting which one will actually work. So the best strategy is to try at least 3 guides to ensure that you're getting the best results possible, because you may be getting low editing simply because of the guide selection. The other thing that we can try to do is improve the way that the reagents that we're actually using for CRISPR experiments. And so we at Synthego have pioneered a way of synthesizing synthetic, chemically modified single guide RNAs. So these are ones that are chemically modified at the 5' and 3' ends of the guide itself, with the 3 bases at the 5' and 3' end. And what that does is it helps reduce the exonuclease degradation that occurs for the guide, as well as does not trigger an immune response, which other formats may, because they're the RNA that are floating around in the cell. And as you can see here, this also combined leads to an improved editing efficiency in comparison to IVT guides. So we're able to have a huge increase in editing efficiency just from the format. And so talking about format, there's lots of different ways that people have made CRISPR reagents. And so there's been a very big evolution in those reagents. So at the very beginning, a lot of people use plasmids to introduce the CRISPR reagents, so the Cas9 and the guide RNA. This required cloning into a plasmid and preparation. And unfortunately, a lot of these plasmids had cytotoxicity because they were large pieces of DNA that the cell just didn't really like. They also could integrate into the site, which when we're doing precise genome engineering is obviously not ideal. And one of the biggest issues that plasmids have is that they have a long half-life. So what that means is when we overexpress Cas9, for example, using a plasmid, we often still see Cas9 expression even 72 hours after editing has occurred. And so what that means is that even though we've already made the double-stranded break and the cell has repaired it, that Cas9 and guide are still floating around and can potentially have other off-target effects. And so many people have now demonstrated that using a plasmid approach leads to much higher off-target effects. And again, you can't chemically modify them, the plasmids, so we aren't able to improve the editing efficiency just through those kinds of modifications. You can also make your own guides using in vitro transcription. This usually is very labor intensive, as well as usually carries along a lot of impurities because this is an in vitro way of synthesizing guides. That leads to lower editing efficiencies. And again, you can't chemically modify them to then improve those editing efficiencies. There is synthetic chemically modified guides that come in 2 pieces, and this is called the 2-piece or the CRISPR tracer RNA guides. They are synthetic and they are chemically modified, but because you have to do a pre-annealing step before you anneal it to the Cas9 to make the ribonuclear protein, this leads to inefficient annealing and thereby inefficient editing as well. And so lastly, what we would recommend, especially for hard to transfect cell lines, is to maximize your editing efficiency to use the synthetic single guide RNA that's chemically modified. So these are very pure, they're very well tolerated by cells, and especially for hard to transfect cells. And because we're able to chemically modify them and they're transiently expressed in the cell, so they have a lower half-life, but still get very high editing efficiencies. So the germline effective editing is still present, and we get reduced off-target effects. And when we've actually compared the 2 different formats that are synthetic and chemically modified, so the CRISPR tracer and the single guide RNA, we do see that we can improve editing efficiency even further. So this is data where we looked at nearly 40 different guide RNAs using both the CRISPR tracer from ourselves and different vendors, as well as our synthetic single guide RNA. And so what you can see in this is that for some guides, you're always going to get low editing efficiency. And this is likely to be an influence of the sequence itself rather than the format of guides that we're using in this experiment. There's also going to be instances where the editing efficiency doesn't really change depending on what format we use. And so this, you could easily use a CRISPR tracer for this. However, what we most commonly see is that you get a decrease in editing efficiency depending on that format. So using a synthetic single guide RNA gives you the highest editing efficiency, and then with every different format that we use, we're losing editing efficiency. So we're still getting relatively good with our CRISPR tracer, but with other vendors, they're not usually as high quality, and so we lose editing efficiency for that. And so one thing that we would recommend is using synthetic single guide RNAs, because that ensures that you're always going to get the highest editing efficiency possible. So once you have your guides, how do we then actually utilize those in our experiment? And one thing that I would always recommend is optimizing the way that you're transfecting. And if possible, we recommend electroporating your guide and Cas9 into the cell as an RMP, because that will usually give you the highest editing efficiency. And we usually see about 2/3 reduction in editing efficiency if we use lipid-based transfection. And one thing that we at Synthego do is we really embrace our ability to fail, which, as this quote from Thomas Edison very nicely depicts, in general, scientists usually are very good at failing, and it's not really a failure, it's just finding a lot of ways that don't work. And so what we try to do is really identify the best conditions by letting ourselves find ways that don't work. And so this is really nicely depicted with how we actually automate our optimization process for engineered cells. So any cell line that we take in to do an engineered cells product or project with, we test 200 different conditions for that optimization. So for example, when we did this in THP1 cells, we tested these 200 different conditions, and using the manufacturer's protocol, we would have gotten about 7% knockout efficiency. So this knockout score is actually a score of the insertions and deletions that are going to cause a frame shift, rather than just all insertions and deletions, because there might be some that won't. And I'll touch on this in a second and how we determine this. However, you can see here that when we were able to optimize, we were able to go from 7% all the way up to 72%. And so this is a huge improvement in the editing efficiency. And because we have this automated and high throughput process, this took us only 3 days. And this really, most of that time was in waiting for the results to come back. And so we do this for every single cell line that comes in, as I mentioned. And so this is just some of the cell lines that we've been able to optimize and the high knockout scores we've been able to achieve with these cells. So once you've actually done your experiment, how do you analyze it? And so historically, there's been really two different ways of analyzing CRISPR experiments. One has been using the T7E1 or surveyor assays. And so how these assays function is if you make a double-stranded break with CRISPR, you're able to, once you PCR amplify around that region, you'll get an amplicon of that double-stranded break, which will have mismatches to its other template when it's PCR amplified. And then we can use a nuclease to be able to digest at those mismatches, the T7 nuclease, and you can resolve that on a DNA gel. So this is very qualitative, obviously very time consuming. So the other way that people were doing was with amplicon-seq. So this is where you would PCR amplify around the cut site and then send that in for next-gen sequencing. So how do these two methodologies actually compare? So one's obviously very qualitative, NGS is very quantitative, but very laborious. And so unfortunately, these two techniques don't really compare to each other. So this is data generated by Chandra Miller-Pruitt, who runs the WashU Genome Engineering Core. And what she was able to show in this publication that was published this year was that when she compared next generation sequencing results to T7E1, there was actually no correlation between the two. And worst of all, in every case, T7E1 vastly underrepresented the actual editing efficiency that you got. And at the worst situation, you would actually, the T7E1 would show you that you got 10% editing when actually you were able to get 90% editing. And this could really change how you do the next steps and the kind of work that you need to put in. And so we obviously wanted a method that was easier to do than next-gen sequencing, but a lot more comprehensive and more accurate than the T7E1 or surveyor assay. And so what we decided upon is kind of the middle ground of the two. So what we do internally and for any engineered cells project is we PCR amplify around the cut site, and then we send that in for Sanger sequencing. And we've developed our own software tool that is able to deconvolute the chromatograms that come out of Sanger sequencing to give us a relative view of the kind of insertions and deletions that the CRISPR-Cas9 has created. So we call this ICE, or Inference of CRISPR Editing. It is available from our website, so you just click on Bioinformatics Tools and ICE Analysis and you're able to access it, or just go to ice.synthego.com. It has free batch uploads, and all of ICE is completely free. It has an automatic window that will set the ideal settings for your experiment. And then you get publication quality results and data. And we're always updating this as well. So I'll talk about that in a second. So this is what ICE looks like. All you have to do is put in your guide sequence and then drag and drop or select your control AB1 file as well as your edited AB1 file, and then hit analysis. You can also do batch upload, and we have in our example files, if you download those, there's examples of how to structure those definition files so that you're able to do batch upload as well if you have multiple samples. And we can accept up to 700 samples if you're doing a huge editing experiment. And so this is what the outcome looks like. So this is the first view that you see. And what the important thing that we show you is the I score, which is the total editing efficiency, as well as the KO score. So this is that score that I was talking about that gives you the percentage of insertions and deletions that are going to cause a frame shift. So it removes any things that are integers of 3 and also includes things that are large deletions. And we can use ICE for multiplexing, which I won't show you, but we do have some great articles on our website with how to use multiplexing for your CRISPR experiments if that's what you are after. And the really cool thing that ICE shows you that's really informative is actually the percentage of each insertion or deletion that is present in that population that Sanger sequencing was able to detect. So as you can see from this example, the highest contribution that we have in this population is a -2 deletion. That's in about 68% of that population. So this is really informative for future experiments that we want to do down the line, as well as if you wanted to take these cells and clonally expand them and make a clonal cell line. And when we compare ICE to other techniques, such as TIDE or next-gen sequencing using amplicon-seq, we're able to get very good correlation between these techniques. And we have published a manuscript on bioRxiv if anyone is interested, which can be very easily found if you want more details. And this software is also available on GitHub if anyone is so inclined to dig around at that code. So once we have an ability to analyze these results, how do we put all of this together to give you the best flexibility for your CRISPR experiments? And obviously, CRISPR is it's not an easy tool to set up in your lab. It does seem relatively simple, but there's a lot of things that you have to consider, including all of these things that I mentioned and others. And so as a company, what we wanted to do is provide a solution that gives people the maximum amount of flexibility to be able to utilize the power of CRISPR in their work. And so we have 3 different product lines that depict engineered cells, which the goal of which is to really provide unprecedented access to this fantastic CRISPR technology. So the first is knockout cell pools, where we make a polyclonal population of cells for you, and I'll explain that in further detail. We can also take those to clonal. And then we have an advanced cell category, which includes knock-ins and other complicated editing experiments. So for knockout cell pools, we can quickly and successfully generate these cells where we design and synthesize the guide RNA, we edit those cells, and then we send you a quality control report. And you get 2 vials of knockout cells, 2 vials of wild-type cells, and then a product summary. And depending on the doubling time of the cells and how well they survive post-nucleofection, this takes between 4 to 10 weeks. And so what you get is guaranteed protein knockout. And so we have very, very strong guarantees for these products. This includes if we don't get at least 50% editing, we will either redo the experiment or go back to you and decide what we should do next, but we won't charge you for it. In addition, if you get the cells and you test them in your own lab and you don't see a 50% reduction in protein within 48 hours, we will also go back and either redo the project or refund your purchase price. In addition, we also have a viability guarantee. So this means if we conduct your knockout and due to the biology of the gene that we've knocked out, it's integral to cell survival. If we see a reduction in the knockout efficiency due to that and it goes under 50%, we will also refund your money. So we're really trying to provide no risk CRISPR knockouts. You also obviously get the report, including the insertions and deletions through ICE, as well as primer sequences so that you can redo that ICE analysis once you have the cells in your lab. And then a certificate of analysis showing that the cells are free from mycoplasma, as well as a visual sterility test. Now, one question that we often get is, well, what really are these pools of cells that we're getting? And whether these are required to go clonal. And oftentimes, because using our automated platform and our amazing synthetic single guide RNAs, we're able to get such high editing efficiency that these pools could be useful without actually having to go clonal, which saves you a lot of time and potentially money. So these pools of cells are obviously polyclonal populations which contain different insertions and deletions. And the vast majority of these are likely going to be ones that cause a frame shift because they're not integers of 3. So all of those different insertions and deletions are going to cause a protein knockout. And so if we have a high enough efficiency pool, and we're generally being able to generate pools that are about 70% editing efficiency, so very high editing efficiency pools, these can often be used directly for assays. Or if we go clonal, we can often get very great success rates with clonal as well. And so, for example, this is a product that we shipped to a customer where we were able to get very high editing efficiency. And when they looked at protein expression using flow cytometry, as you can see in the figure on the right, they were able to get the protein expression in the knockout cells was almost the same as the negative control and almost a complete protein knockout in comparison to the wild type. And so if this protein was responsible for a functional phenotype, these cells would very strongly show that phenotype because we've been able to completely knock out the protein function. In addition, there's obviously cases in which you have to go clonal, and we would recommend this if your knockout is causing some sort of population drift, where your editing population is being reduced because the cells are proliferating much slower, or they're undergoing apoptosis in the condition of the wild-type cells. And so in those cases, it's also easy to go to a clonal population using that highly edited pool. And so, for example, if we were to take our pool with high editing efficiency and plate those into single cell plating using 0.5 cells per well, using one 96 well plate, we're able to screen about 29 wells. So this is the number of wells via probability that we're going to get only one cell per well and not 0 or more than one. And so of those 29, because of that 80% editing efficiency, we're able to see at least 22 edited clones. So this is a really large number of clones for not much work. On the flip side, if you were to do this yourself, for example, using a 20% edited pool, you would need to screen 4 96 well plates, which would mean 343 wells that need to be imaged, need to ensure that they're single cell, need to be media changed, expanded, split, genotyped. And of those, you'd only get 6 edited clones, in contrast to the 22 that you get with only one 96 well plate if you used our cells. And in addition, we provide a limiting dilution and clonal expansion protocol that gives some of the tips and tricks that we use internally to try to get better clonal expansion outcomes. So we highly recommend following some of those tricks, and this is freely available on our website on the resource page. The other question we often get using knockout cell pools is, well, how many alleles are actually edited in these cells? And so although we cannot see this in a pool because there's lots of different insertions and deletions that may contribute this, when we take those pools to clonal, we're able to see what that kind of, what the homozygosity or heterozygosity of the edit was. And the answer is, because we have such high editing efficiency, we're able to see that all alleles in general are edited. And so from you can see here, we're able to see really high editing efficiency, and all of the alleles are edited, even in an aneuploid cell line. And so it's actually more difficult for us to get a heterozygous edit, although we can do it, just requires screening more clones. And so we're able to very confidently say that the majority of the editing that we're able to do, because of its so high editing efficiency, will result in homozygous knockouts. So for the knockout cell pools, we have over 700 cell lines available. These are all human cell lines, and they can ship in as few as 4 weeks if you wanted to do a knockout cell pool with us. All you have to do is go to our website and type in the cell line and the gene name. If both of those get filled, you can literally just add to cart, generate a quote, and be able to submit a PO against it. It's as simple as that. Of course, if you do want to talk to us about the process, we're always happy to talk as well. And again, these are all guaranteed protein knockout, or we don't invoice the final purchase price. So there's always cases where people would like us to just do the clonal steps for them as well. And so we take a slightly different approach to knockout clonal cells. In this, we use a milestone approach where we do several different stages and consult with you and communicate how we're doing at each step, because this is a longer time frame because we have to go clonal. So the first step is we onboard the cells, where we have to procure the cells often, we have to expand them, and then ensure that actually test that they can get to single cell clonal cell state, because obviously we want to be able to know that to make sure that we can make a clonal cell line at the end of the project. We then edit the cells, where we optimize, we make the guide RNAs, we then transfect and then analyze that pool of cells, which we then take into single cell clonal expansion. And as you'll see, this takes the longest period of time, anywhere between 5 to 12 weeks, depending on how well the cells grow out of clonal, from that clonal dilution. The other thing that we do at this step is we image the cells every couple of days to ensure that we're tracking the cells and that we're only getting a single cell clonal population in each well. And we obviously discard any wells that are coming from 2 cells. This ensures that we're able to screen clonal populations, and then we also validate this during the validation step in our quality control to ensure that the cells that we're expanding and growing up for clonal delivery are only monoclonal cells and aren't a result of 2 potential clonal populations coming together. And when we have generated some of these, we're able to show very strong functional outcomes because we have clonal knockouts. So in this example, we took C2C12 cells, which is our myoblast cell line, and we knocked out the MyoD1 cell. Now, MyoD1 gene, excuse me. And so these cells are ones that can differentiate from a myoblast into a myotube. And you can see on the images here on the right, when we have no guide RNA and we've differentiated these cells, they're able to very effectively differentiate into these myotubes. But when we block MyoD1 by knocking it out, which disturbs that differentiation process, we're able to see that we now no longer have myotubes being formed, as you can see in that third panel on the right. And so this really does depict that we're able to find functional clones that have a functional phenotype, which is obviously what you're looking for in using CRISPR as a resource for your protein knockouts. So our knockout cell lines are available again in those 700 cell lines, immortalized human cell lines, and the turnaround time is longer because of that clonal process. The price point is also higher at $15,000 per clone, and this is really attributed to how long it takes to be able to get that clonal cell line. Again, it's completely guaranteed, and so if we aren't able to get you that clone, we will give you your money back, and we will work with you at all of the steps to ensure that we're getting the best product possible. So obviously this is a very high price point for a lot of researchers, and so this is why we have really tried to make knockout cell pools a more viable option for a lot of people, where you can either get a functional pool and be able to use that directly, or take that to clonal yourself. Because of the high editing efficiency, this is so much simpler than trying to do this yourself. So finally, we have advanced cells. So these are cells in which encompass other editing experiments that are more complicated. So this includes knock-ins as well as other mammalian cell types that aren't human. And so in advanced cells, we're able to generate these more complex cell lines. So this includes designing and synthesizing both the guide as well as the donor template if we need to utilize a donor for a knock-in. We then edit the cells, isolate them, and do clonal quality control testing. And so all knock-in experiments are currently delivered as clonal cell lines. What you get is 2 single-cell clones as well as a project summary, which is also detailed during that milestone process. So we also use that milestone process for knock-in clonal projects or advanced cell clonal projects. This includes all of those other steps, including onboarding those cells, checking for single-cell clonality ability, designing and making all the reagents, editing them, then expanding them into single-cell clones, and doing that final QC and validation. And so advanced cells encompasses more of those complex editing reactions that usually are quite difficult for people to attain in the lab, because as I mentioned at the very beginning, homology-directed repair, which is the mechanism by which we do knock-ins, is much, much less preferable by the cell. And so getting double-digit knock-in editing efficiency is quite frankly phenomenal, and we're able internally to be able to get anywhere between 10 to 20% knock-in efficiencies, which is amazing. And this is because we're able to optimize so many of these processes in a high throughput way. So these advanced cells include things like SNP changes, codon changes, tagging genes, including GFP, as well as other tags, both biochemical and fluorescent tags, as well as insertions, and includes some of those more difficult immortalized mammalian cell lines, including mouse and rat cell lines. And we can deliver both clonal cell lines in about 10 to 20 weeks. So what I've shown you today is that within engineered cells, Synthego is trying to provide unprecedented access to this CRISPR technology in a very affordable and flexible manner by being able to provide high editing efficiency pools, which can be utilized directly by knockout cell clones, and then advanced cells, which encompasses those more complex editing efficiencies. And we're able to do this by really combining our full stack genome engineering solution. So this includes using the most effective reagents, using those modified single guide RNAs, which give us both the best editing efficiency as well as the lowest off-target rate, including using our design tool, which incorporates all of those design metrics that I mentioned earlier. We optimize our methodology to ensure we're getting the best editing efficiency possible. And we have developed these great analysis tools to be able to make better analysis and more informative analysis in a quicker turnaround time. And then finally, we're able to, because we have such high editing efficiency, provide flexible options that are affordable for all researchers. So with that, I encourage you to really utilize your time in discovering those amazing things that all of you researchers are doing. And we really want to be able to help you in doing this by optimizing the CRISPR for you and being able to generate these resources for your experiments rather than you having to do that yourself. So if you're interested, please contact us at the address below, and we'll be happy to talk you through the options. And in addition, I'd like to give a shout out to our amazing blog called The Bench. This has lots of different stories and really cool things that people are doing with CRISPR. So if you're interested in ensuring that you're up to date with the best CRISPR news, I would highly recommend subscribing to the blog. So thank you so much for your attention. And with that, I will take any questions.
Thank you, Dr. Wronski, for that informative presentation. We will now start the Q&A portion of the webinar and will address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, how do you guarantee that you will get 50% editing?
That's a great question. And so we not just guarantee 50%, we guarantee at least 50%. And so we're able to provide this guarantee because of the high level of optimization and the automated process that we do. So as we do any experiment, we obviously optimize those 200 conditions that I mentioned. We also try multiple guides at the same time, so we're able to pick the best one and not waste any time and lose any time in going back and trying something else. And then because we have this full stack, we're able to see these things really quickly. So we're really very well positioned because of the automated platform that we've been able to build out to be able to deliver these really high editing efficiency. So that really is why we have the power to give that guarantee and such a low price point.
Thank you for that. Next one is, what about off-target effects? Do you assess those?
Yeah, that's a great question and one that always comes up. So obviously everyone is concerned about off-target effects with CRISPR. And the thing that we tend to see is that using both well-designed guides, like we are doing with our design tool, and utilizing the synthetic chemically modified single guide RNA format using an RMP to deliver those cells, we're able to see usually less than 1% of off-target effects and usually much even no off-target effects. We don't analyze those as part of an engineered cells project, but we do give you the guide RNA sequence that we've used, and using our design tool, there's a validate portion on that where you can type in that guide sequence and identify where potential off-target effects may occur. You can then use that information to develop, for example, a Sanger sequencing strategy to be able to identify whether or not off-target effects have actually occurred there. So while we don't test for off-target effects at the moment internally, we definitely provide you all of the tools to be able to do so yourself.
Thank you for that. Looks like we have time for one more question. Do you offer academic pricing?
Absolutely. So one thing that as a company we're trying to do is be much more accessible. And so obviously some of these services as provided by other CRO organizations can cost $10,000 to $20,000. And so we definitely have academic pricing. And if you're interested in that and are from an academic lab, you can actually just ask for academic pricing directly from our website. There's a little button that says, you know, I'm an academic, please give me academic pricing. In addition, we also offer bulk discounting pricing. So if you're interested in doing a lot of projects with us, definitely get in touch with us and we can offer you even further discounts for that. In addition, if you would like to do the editing yourself and use the synthetic chemically modified guides, we also have special pricing promotions on those. So, you know, for whatever your application, it's definitely worthwhile to get in touch with us because we can usually offer some really great promotional discounts to make this more accessible to people.
Thank you. I would like to once again thank Dr. Wronski for her presentation. I'd also like to thank Synthego for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 27th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.